Good morning, students. Today we'll have to discuss about the transportation, certain part of the transportation in human being as well as in plant, and some of the part of the excretion also. We have discussed the transportation in plant by the help of the absorption, ascent of sap, transpiration, like that. But today we will have to discuss about the transportation of food in form of fructose from leaf to different parts of plant. Different parts of plant. This particular system, the food which is prepared by the leaf by the process of photosynthesis, will be transported from the different part of the plant through phloem. Inside phloem, by the process of diffusion or the higher concentration to lower concentration, the food will move inside the phloem, and ultimately in phloem they will be transported from the different part of the plant. Munch has proposed a theory, hypothesis, that is called mass flow hypothesis. It has no so much proof, but there is no any alternatives are also, so it is more or less accepted. What happens according to this hypothesis, we will have to discuss. Suppose this is the phloem part. This one is the mesophilic cells, and both are connected together with the certain tubes. Suppose food is prepared here. Food is prepared, and this food is soluble in form. Then what happens? Due to higher concentration of that particular soluble form, the water start to move in this way. Then what happens? The volume of the solution will increase, and that solution start to move from the one part to the another part, and ultimately it will come here. This movement from this particular part through phloem to this particular part, and this is called mass flow. Now if water will be inserted here to this particular part, then what happens? The water from, in this way there is a circular path, and on this particular circular path the food will be transported, and this is called mass flow hypothesis, and this is proposed by the Munch. It is one of the part of the transportation in plant. Now one another part in human being, that is called double circulation. That is called double circulation. As you know that the human heart have four chambers. Suppose this one is the heart, This one is the heart, and it has 4 chambers. This is the right hand side, this is left hand side, so it is called right auricle, it is called right ventricle, it is left auricle, it is left ventricle. Now what happens in it? You know that the blood will move from different part of the body to this one and through this one and through this one. It is called superior vena cava. Superior vena cava. It is called inferior vena cava, and it is called coronary sinus or coronary vein. Just we have seen that the deoxygenated blood from the different part of body to come inside the right auricle. Now through right auricle, from right auricle through right auriculoventricular aperture, it will move to the right ventricle. It is deoxygenated blood. Just we have make a arrow by green. It is deoxygenated blood. Again, this blood move from this right auricle to the right ventricle. Now the blood will have to move for the deoxygenation, and it will move to lungs. Here is the lungs. Here is the lungs. Now oxygenated blood, the oxygenation will take place here. Then what happens? The oxygenated blood will move to this particular part here. Now again this blood move to left ventricle through left auriculoventricular aperture, and now this blood will move to the different part of the body through aorta, that is called systemic aorta. Just we have observed that the one blood which is deoxygenated will come firstly right ventricle, right auricle, then to the right ventricle, and the same blood will go to the lungs. Now that particular blood which is deoxygenated become oxidized. The removal of the carbon dioxide takes place, the addition of the oxygen will take place, and again this blood will move to the heart. So the twice a time the blood will move inside it, and only oxygenated blood will move to the different part of the body. It is one of the significant characteristic feature of the double circulation, that not mixed, not deoxygenated, but pure in form of the oxygenated blood will move from the heart to the different part of the body. And this type of circulation is called double circulation. Here you will see that the blood from the right auricle to right ventricle to the lungs, that is called pulmonary circulation. From the lungs to the left auricle to the left ventricle to the body, that is called systemic circulation. And one circulation is also here from the coronary aorta. The blood will move to the different part, different wall of the heart, and again that will return back to the right auricle. So that is the coronary circulation. So three types of circulation. One is the pulmonary circulation, another is the systemic circulation, and third one is the coronary circulation. In this way, we will see that the double circulation will take place. This is all about the cardiac cycle. In previous lecture, we have discussed the cardiac cycle, and this is the one of the remaining part of that lesson, that is called double circulation. Good morning, students. This is the one of the part of the excretion in human being, that is called artificial kidney or the hemodialysis. As we know that the excretion is the process of filtration of the metabolic waste from the blood in case of human being, and that is the major type of excretion. Of course, removal of the CO2 is also called excretion, but here we will see that the removal of the nitrogenous waste from the blood is called excretion. Elimination of or the get rid of the metabolic waste from the body. We have discussed in different way how filtration takes place, how the useful filtrate return back to the blood, and how the ADH and RAS system will support this particular excretion or the maintenance of the osmotic pressure. This is a question whenever the kidney is not functioning in a human being. We know that a pair of kidneys is present, but in certain times it has been observed the kidney is not functioning. Both the kidneys are failed. They are not in condition to filter the blood in proper way. Then there is a chance of the increasing the concentration of the urea inside the blood, and after some times the human being cannot survive. So what happens if kidney is not functioning? The one thing, that kidney can be transplanted, but it is not a easy task. It is a very major task, because firstly the kidney is not available. Secondly, if kidney is available, then that is not from the siblings or the others. So there is so many problems. So in addition to that particular transplantation, there is one means for the survival, to make it survival, that is called artificial kidney. How the blood can be filtered without kidney. This is the artificial kidney or the hemodialysis. Now what happens in it, just we will discuss. Suppose this is the equipment where the dialysis will be taken place. Suppose it is equipped with a very much coiled fine tubular structure. Fine tubular structure. This particular part is filled with the This is fine tubular structure, and that is mainly made up of the cellophane membrane, which is permeable, selectively permeable. This part of the equipment is filled with the isotonic solution of the blood. This is filled with the isotonic solution of the blood. And it has been observed there is a continuous flow in this particular tubes. The flow of this isotonic blood is like this. It has all the components of the blood except urea. Urea is not present in it. Urea is absent. Urea less. Urea in minus. Now what happens? The blood from the patient, the blood capillaries are attached with this one, and firstly the blood will be added with the some anticoagulant to prevent the coagulation, and the temperature is also low to prevent the contamination of the blood. Okay? Blood will be taken from the human being, that will be mixed with the anticoagulant, and continuous flow. The whole blood will not be taken one time from the body, but there is a continuous flow of the blood, and that will be added with the anticoagulant, and the temperature will be low for the prevention of the microbial infections or others, and that will insert inside it. Here we will see that it is urea containing, and here urea is not present. And it is selectively permeable. It means the urea start to come out. Urea start to come out. Urea start to come out. In this way, you will see that the continuous flow of the urea from this particular tubular to this particular liquid. It is the liquid of the certain salt, which is isotonic to the blood. Now, due to increasing the percentage of the urea, a time will come that the urea of the blood capillary and urea of this part become equal. So the movement of the urea is stopped. So because to prevent this particular, what happens, that there is a continuous flow of this liquid and the drop wise storage will be takes place. So this condition will never come, that the urea concentration will never equal to that of this blood and this one. And a time will come that this urea inside blood is up to mark, and then it is filtered blood. Filtered blood. Now before giving inside the patient, what happens? The anticoagulant will be released. The temperature will be equalized, and ultimately it will be gone, and now the human being will not suffer from the urea problem. This is the artificial treatment.